Good morning, students. Today I am here with third and last part of Biomolecules of 11th standard. Here in this session we are going to discuss about enzymes, nomenclature of enzyme, classification of enzyme, mechanism of enzyme action and factor affecting enzyme action. So let us start the session with enzymes. Enzymes are organic compounds necessary for normal metabolism in the cells. They are proteins that catalyze the biochemical reaction. So they are also called as biocatalyst. Enzymes are very specific for their substrate and they require optimum temperature and pH for action on the substrate. It helps in enhancing the rate of reaction by decreasing activation energy. Now let us discuss about nomenclature of enzyme, that is naming of enzyme, how the enzymes are named. Enzymes are named generally by adding the suffix ase, A-S-E, to the substrate. For example, sucrose is the substrate and sucrase is the enzyme. Like that, lipase, proteinase, nuclease, etc. It is also named according to the physiological activity it catalyzes. For example, oxidase, polymerase, dehydrogenase, carboxylase, decarboxylase, etc. Some enzymes are also named the source from which they are obtained. For example, the enzyme papain is obtained from papaya. Now it is about classification of enzyme. Enzymes are classified into 6 different types. They are Oxidoreductase, Transferases, Hydrolases, Lyases, Isomerases, Ligases. So we will discuss one by one. First is Oxidoreductase. Oxidoreductase enzymes are responsible for catalyzing oxidation-reduction reaction by transfer of hydrogen or electrons from one compound to another compound. Oxidases enzyme complete oxidation reaction by addition of oxygen to a substrate or by removal of hydrogen or one or more electrons, whereas reductase enzyme catalyzes reduction reaction by removing oxygen or by adding hydrogen or one or more electrons. In the given reaction, it is shown addition of oxygen to beta D-glucose to form D-glucono-1,5-lactone and hydrogen peroxide. And this reaction is catalyzed by glucose oxidase enzyme. In this, there is conversion of ribonucleotides into deoxyribonucleotides with the enzyme ribonucleotide reductase. Second enzyme is transferases. With the help of this enzyme, there is transfer of a specific group other than hydrogen from one substrate to another. In the given reaction, you may observe that there is group transfer with the help of alanine transaminase. When alanine and alpha ketoglutarate is combined, then the enzyme alanine transaminase catalyzes it and the product will be pyruvate and L-glutamate. Third enzyme is hydrolases. This enzyme catalyzes splitting of larger molecules into smaller molecules by addition of water. Here you may see that pyrophosphatase is the example of hydrolases enzyme. which catalyze hydrolysis reaction where water is the acceptor of the transferred group. In the given reaction, pyrophosphate is converted into phosphate by addition of water. Other example of hydrolases are esterases, peptidases and glycosidases, which also do the hydrolysis reaction. Another class of enzyme is lyases. Lyases catalyze the breaking of specific covalent bonds with removal of groups without hydrolysis. Here there is no addition of water. Here pyruvate decarboxylase, which is the example of lyases, catalyzes pyruvate into acetaldehyde and carbon dioxide. Here pyruvate is the substrate, whereas acetaldehyde and carbon dioxides are the products after enzymatic activity with the help of enzyme pyruvate decarboxylase. Now it is about isomerases. This enzyme helps in rearrangement of molecules in the substrate into a related isomeric form. So thus there is change in the structural formula, but molecular formula remain same. In the given reaction, you may observe that malate isomerases catalyzes intramolecular transfer of groups, and malate is changed into fumarate. You can see here that only there is rearrangement of molecules. Other examples of isomerases are inositol 3-phosphate synthetase and maltose epimerase. Now the last class of enzyme is ligases. This enzyme catalyzes the joining of two substrate molecules by getting energy from hydrolysis of ATP, that is adenosine triphosphate. Here you may observe glutamine synthetase is responsible for addition of NH4 to L-glutamate, and finally L-glutamine is formed. Here you may see that there is hydrolysis of ATP into ADP and inorganic phosphate, that is PI. ADP is the adenosine diphosphate. Now let us see about mechanism of enzyme action, means how enzyme works. Whenever we revealed the three-dimensional structure of enzyme, then we find that there is one or more active site where the substrate binds. It is based on lock and key hypothesis. The active site on the enzyme acts as a lock, whereas substrate which fits into it is like a key. The point where substrate bind is called substrate binding site. When the substrate bind to the enzyme, then there is lowering of activation energy and the reaction will produced at a faster rate. When the substrate binds to the active site of the enzyme, then there is change in the shape of the enzyme so that substrate fit more tightly. As a result of it, there is breaking of chemical bond of substrate and formation of enzyme product complex. Further, enzyme releases the product and enzyme is free to take up another molecule for enzymatic action. So, in the whole mechanism, we have seen that enzymes do not take part in the chemical reaction, but they speed up the rate of chemical reaction and also lowering the activation energy. In the given graph, you may observe the amount of activation energy without enzyme and with enzyme. With the help of enzyme, activation energy is less, whereas without enzyme, activation energy is more. Now let us see about the factors that affecting enzyme action. Temperature, pH, concentration of substrate and chemicals are the common factors that affect enzyme action. We will discuss in detail one by one. First of all, we will discuss how temperature and pH affect enzyme action. Enzyme always works in a narrow range of temperature and pH. Enzymes are more efficient at optimum temperature and pH. Optimum and temperature and pH means it should not be too less or too high. Enzyme activity lower down when the temperature and pH are below and above optimum. Low temperature preserves the enzyme in a temporarily inactive state, but high temperature destroys the enzyme action. Why? Because enzyme is a protein and high temperature denatured the protein. Most enzymes show maximum activity in a temperature range of 25 to 40 degree. The optimum temperature of an enzyme usually corresponds to the body temperature of an organism. Most enzymes show maximum activity in a pH range of about 6 to 7.5, that is near neutral pH. In the given graph, it is about the effect of temperature and pH over enzymatic action. Now the next factor which affect enzyme action is concentration of substrate. When the concentration of substrate increases, then the velocity of enzymatic reaction rises at first, but ultimately it reaches a maximum value, that is Vmax, maximum velocity. And further, there is no change in the velocity by the rise in the concentration of the substrate. This is the graph which shows relation between substrate and velocity of enzymatic reaction. Another factor is chemicals. Enzymatic activity is also sensitive to presence of specific chemicals that binds to the enzyme. When binding of chemicals to the enzyme shut off the enzymatic activity, then it is called inhibition. And that chemical which shut off the enzymatic activity is called as inhibitor. Whenever the inhibitor is closely resembling with the substrate in its molecular structure and inhibits the enzyme activity, then it is called as competitive inhibitor. For example, inhibition of succinic dehydrogenase by malonate, which closely resembles the substrate succinate in the structure. Now we will discuss about cofactors. Cofactors is a non-protein chemical compound which tightly and loosely bind with an enzyme or any other protein molecule. The protein portion of the enzyme is called apoenzyme, and for any enzymatic activity, cofactor is essential. Thus, it is considered as helper molecules. There are three kinds of cofactor. They are coenzyme, prosthetic group, and metal ions. So let us discuss about this. Prosthetic groups are organic compounds that are tightly bound to the apoenzyme, whereas coenzyme is an organic non-protein compound that also binds with an enzyme to catalyze a reaction. Co-enzyme can be reused several times when bound with an enzyme. For example, NAD and NADP. Co-enzyme cannot function alone. Some metal ions like iron, magnesium, manganese, cobalt, copper, zinc, molybdenum are also required for several enzymes for its activity. So this is end of today's session. Thank you. For any kind of doubts, please write your question in comment section.